So interesting enough, the name Warfarin refers to the Wisconsin Alumni Research Foundation, who sponsored the studies on this, at the time, product of killing rodents. And why is this important? Rodents were always competing with our own food sources because we keep large storage of food, and mice and rats can take advantage of that. And it's not just the fact that they're eating our food, but the fact that they're contaminating our food led to people to produce this particular chemical that were causing internal hemorrhaging in rats. It turns out that within a short period of time, so let's say decades, because of already existent variants, so mutations in rats that can survive this anti-clotting chemical that is found in warfarin. All the rats that were susceptible to this rodenticide died or eliminated, and then the next generation was fast forward evolving because of the allelic frequency of the resistant allele predominated in the future generations. Very similar to what happened with humans in the medieval time when the bubonic plague eradicated the individuals that were more sensitive to this particular bacterium that causes bubonic plague. Obviously, there are no antibiotics developed at the time. So about a third of the human population was eliminated. So we're all now offsprings of the ones that had that particular gene. So since then, we're not using this anymore because it's not working on this generation, on this new populations of rats that have more abundant representation of the resistant allele. And the same thing is happening with the use of antibiotics, either in farms of overcrowded individuals, farm animals, that are given antibiotic as a preventative. So they're not even sick yet, but they're given antibiotics to prevent sickness because they live in very close quarters, because it's very expensive to allow them to live more naturally, and so on. All of that leads to selection and slowly microevolution of individuals that are not even immune, that are not even susceptible to that particular antibiotic. So even though the antibiotic used to kill the particular bug that we were interested to kill before, now it doesn't. So another very clear example is MRSA, or methicillin-resistant Streptococcus aureus. So Streptococcus aureus, it's a relatively common bacterium that now acquired this allele. Well, actually, it's not an allele because a bacterium has only one, usually circular chromosome, but it acquired this mutation, this gene that can gives it resistant to methicillin, which was the ultimate resource to killing this particular bacterium. And this is just one example of a superbug. Clostridium difficile or difficile, it's another example, and so on. So every time we use a very strong killer of anything, we're actually selecting for stronger individuals, or for the very few individuals that have the mutation that allows them to have a differential survival and next reproduction. So very clearly to understand, individuals do not evolve. So even though we change through our life in a various way, and we hope we grow and become wiser and so on, we don't necessarily evolve because we don't acquire any heritable traits, as Lamarck was mistakenly thinking. But the population itself evolves by selecting for the individuals that are better adapted to that environment at that particular moment in time. So mutations are the major source of new alleles, New alleles means new potentials for survival of different new changed environments itself. Even though bacteria do pretty good for themselves in evolving, and in particular to our demise, evolving resistant to antibiotics and everything we throw at them. The reason that they do so good and they evolve so fast is because they have a very short generation time, and that allows them numerous iterations of chances for change, so chances for new mutations. And they also grow in very large populations according to the resource they are depending on. But as you again read in your Making of the Fetus additional book, sexual reproduction, so for example, rock pocket mice that you read about or you will soon read about, can promote one particular allele in one habitat better than the other. So that brings me to another question of review that I would like you to look into it. The question is, what is a population? And of course you have the answer here, but which one is larger? Which one is a nested hierarchy of the other? It's the population bigger than a species, meaning that a population includes several species, or is it the other way around? So a species has different populations spread out in different locations of the Earth. So on this empty slide, I want you to quickly review a few concepts that you hopefully learned in previous classes. If not, just look them up. So I want you to compare and contrast the concept of gene versus allele, and then versus locus. Okay, so how are they different? Are they the same thing? And then how much the same thing, right? So locus comes from Latin and refers to the location of that set of nucleotides or succession of nucleotides that controls the succession of amino acids in a protein. But what is an allele versus the gene? And how many alleles per locus do we each have for every trait? And how many alleles per locus does a population have for each trait? So it turns out that the word allele comes from different in Greek. So it's a different gene or a different variation of the same gene controlling the same trait. And as I said, this comes from location, or the location of a particular gene on the chromosome. And you already know now that there are multiple genes per each of the chromosomes, and that every species has a particular very clear number of chromosomes. I also want you to review very quickly the idea of homozygous, which means the same zygote. In what way the same, versus heterozygote. Heterozygote, right? So different zygote. So does it have the same alleles or two different alleles? How many alleles per locus can each individual have, right? Two, because we're diploid. And then also the idea of dimorphism. And that can be more complex than what I want you to go for here, versus polymorphism. So two shapes versus many shapes. What is that referring to? So within a population, you can have only two alleles, or two different alleles, meaning that you can only have individuals that are blonde versus brunette, and there's nothing in between. Or you can have peas with just green seeds versus yellow seeds. There are no others, purple or light green or dark green or orange and so on. Or you can have blue eyes versus brown eyes and so on. So, or you can have a population that has many shapes or many variations at the same locus, by having several different kinds of alleles per locus within the population. We know that each of us can only have two alleles, the most. And there are even cases in males, in humans, where they can have only one allele per one locus if that allele is on the X chromosome, or the Y chromosome, of course. So an example for polymorphism in human population would be the ABO blood types. So an individual can have only two of these three alleles, but for the whole population, you can have four different kinds of blood types, depending how which of those potential three types of alleles can you get. You can get an A and a B, and then you're an AB, or you can get only an A and an O, and then you're A, and so on. Okay? So these are concepts that you need to have in your active vocabulary. You need to be able to understand really quickly every time you read them. And if you don't, all you need to do is just put more effort into it. So give me an example of a locus for which you are homozygous to, and you're convinced that you're homozygous for that particular locus. And give me an example of a locus in your genetic makeup that you know for sure that you're heterozygote, and convince me why is that the case.